Thank you very much, Doctor. Well, I think it is a great disadvantage to be the last speaker. Many of you are very tired. I think I'll try to go as fast as possible and finish it as early as possible as well. I've given the title as toxicants. Toxins or toxicants are the same in plant foods and water resources, but I'm going to speak about only two of them and not all the toxicants in plants and foods. Now, toxicology, I think, has existed from time immemorial. Ancient civilizations knew very well that nomads to hunter-gatherers what was edible and what was not edible. From about 350,000 species, only 3,000 are currently being used and less than 300 are domesticated. That is the agriculture products. Over time, what is safe, unsafe, processing, shortage, storage, etc. have been delineated. Methods have been devised, toxins characterized, antidotes prepared, and the subject of toxicokinetic and dynamics has evolved to such a great extent that we continuously talk about risk-benefit analysis. Well, this is in brief about toxicology. Today I propose to speak on Lathyrus sativus, which perhaps you may all be wondering whether the disease exists still. It does, but it in pockets, not as in epidemics. But what is important today is, both in relation to Lathyrus sativus and fluorine, I wish to discuss whether they are friends or foes. There is a Sanskrit saying which says, Mane adichetasi amritam api visham bhavati. It means if you take beyond measure, even nectar may become poison. This is what Paracelsus said ages ago, that the dose makes the poison. This is true for both Lathyrus sativus and fluorine. This is the Lathyrus sativus plant. It is a leguminous plant. It is a hardy crop, drought-resistant crop, and requires very little of water, and therefore usually grown during drought seasons. And the disease was prevalent when this particular substance was taken more than 30% in the diet. It has existed from ages. Even Hippocrates described it. Reports exist from 18th, 19th and 20th centuries, that is during post-war periods. It's a survival food, and it's called Lathyrus sativus or Kesari dal in our language. High yielding, drought resistant, low water requirement. Consumption of these seeds results in neurolathyrism, an upper motor neuron disease characterized by spastic paraparesis. And several stages have been documented. It is very insidious in onset, occurs when the seeds are consumed in amounts ranging from 200 to 400 grams per day for more than 2 to 4 months. That's a clinical picture. This is the dal made out of the kesari, the pods which come out of this Lathyrus sativus plant. These are the rotis made out of the Lathyrus seeds, which contains very little of wheat but mostly of Lathyrus seed powder. And this is taken without any diversification of the diet, and that's the reason you get the disease. But it is a savior from the point of view of looking at the proteins and the total dietary fiber, carbohydrate. It also has vitamins, though not to the same extent in other pulses. It does have. So it is a savior during drought seasons. It's consumed in India, Nepal, Bangladesh, Ethiopia, and in many parts of Africa, Somalia, etc. And the last epidemic was in Ethiopia in somewhere in 1997. In India, Bihar, Uttar Pradesh, Madhya Pradesh, Maharashtra, Punjab, West Bengal, Andhra Pradesh, Karnataka and Chhattisgarh have reported lathyrism. Occurs when consumption exceeds 30% of the total diet. It has also been used for adulteration. Now, the toxin in the seed is what is called as a beta-oxalyl alpha-beta diaminopropionic acid, commonly known as ODAP. It's also called BOAA. Well, I've already talked about the paralysis it produces, and pathologically it's a bilateral atrophy of the distal pyramidal tract and spinocerebellar tract and anterior haunches. Sphincters remain normal. The incidence varies during drought anywhere between 2 to 4%. You would note this, the percentage is quite small. But unfortunately, large-scale epidemiological studies in different states of India have not been done. The disease has almost disappeared currently. So you may be wondering why I am presenting laterism today. I want it to be known as a friend rather than as a foe. I'll come to that point later. It's a poison of the mitochondria leading to excess cell death. It's an excitatory toxin leading to excitotoxic death. The good part of the toxin is it's readily soluble in water and can easily be leached out. Based on this, prevention steps have been given to both at the home level and the community level. Moist heat, boiling, steaming, it denatures even the protease inhibitors which are present in the pulse, because that depletes the protective sulfur amino acids. Toxins are excreted in urine and fecal matter. It is not metabolized in animals, but in man, the current studies using radio-labeled studies show that it is metabolized very well in human subjects, and that's the big gap which we do not know. When the humans are capable of metabolizing ODAC, why is it that they are having the disease? Probably it's because their detoxification capacity is limited for this particular toxin. Varietal differences exist, ranges from 0.1 to 2.5%. Well, of course, I don't want to go into the animal studies in greater depth. All that I can only say is none of the laboratory animal model has all of them are a far cry from what the clinical picture you encounter in the human beings. So really we do not have very good animal models, and that's the reason the disease has never been studied in depth, including the pathogenesis, though currently in vitro cell systems several mechanisms have been studied. Well, primary studies are the ones which have been done by both NIN and as well as the Frederick Institute in US. And the animals have been made acidotic, and then only the toxin enters the brain. And young animals are susceptible, but old animals are not susceptible because there is supposed to be a blood-brain barrier. But unfortunately, currently the labeled studies indicate the presence of toxins in the brain irrespective of the acidotic state. So that means there is something which we still do not know about the toxin very well. Now, there are several procedures to remove toxin. Why is this important? Because if you want to use Lathyrus sativus as a friend or as a functional food, then there is a need for this as well. Very simple procedures earlier have been advocated at a domestic level. There is no increase in the cost of food, no radical changes in food habits, acceptable and usable in the culinary preparations. Simply cook the pulse in excess water and drain the water. About 20 to 80% of the toxin can be removed. Or steep the dehusk seed in hot water for 2 hours and dry them. Or soaking the seeds in cold water overnight has also been found to be extremely useful. Parboiling, similar to rice, which conserves vitamins as well, but it has to be done on a commercial scale. And this was advocated in some of the states, particularly Rewa districts of Madhya Pradesh and other places. This was advocated, and experimental models were created earlier. The preventive measures, of course, it's social, economic, agricultural, administrative measures are needed. All this has been practiced in India, and to a great extent the disease has disappeared because there exists a ban on the sale of Lathyrus seeds and flowers since 1955. This is the 45A, PFA rule 45A of the PFA Act. Once it is not sold in the market, naturally the production has come down. And of course now earlier the laborers used to be paid in kind and not in cash, and that was another reason why they had to consume it in large amounts. Now of course they have curtailed the excess consumption. Of course, providing access to other foods and diversification is important, but it is not easily practiced. Genetic engineering, that is promoting low toxin strains using breeding technologies, is really good. And that's what we should attempt to do because the low toxin strains are available. There can also be breeding technologies. Self-pollination of low toxin strain to encourage and avoid outcrossing of this pollination is extremely important. Then you can produce the low toxin strain and grow them. Otherwise there will be admixture and again the plant will have high toxin. Certain agronomic practices like, application of zinc will bring down the toxin content. Alternating with the chickpea in the farm system would also decrease the toxin. And the low toxin varieties have been identified in several countries: Australia, Bangladesh, Bulgaria, India, Kyrgyzstan, Nepal, Poland, Turkey. So many places they have been identified. In some of these places it is grown, but I am not sure whether in India it is still grown despite the ban, but in smaller quantities, whether it is the low toxin or the high toxin strain. I am not very sure about it. Some of the recent thoughts in LS. It is a very critical appraisal done by Dr. S.L. and Rao here, and of course others at NIN in the recent past. They have tried to review the whole situation and try to see what are the newer angles to the Lathyrus sativa seed. In fact, it's now called a golden pulse, which is something very surprising. From a toxin to a golden pulse is really something very interesting. Is it a killer or a savior? I think during drought season, it did save millions of lives, though it did produce the disease. But now there are methods to overcome this high toxic variety of Lathyrus seeds, and therefore perhaps it can be useful. And the causal relationship has been established only when large amount of the seeds were taken. But if it is a small percentage of the whole diet, less than 10%, like 50 grams just as any other pulse, it's unlikely to produce the disease. Even then, the incidence rates were only about 2 to 5% during drought season. Unfortunately, countrywide studies are not available because in those days epidemiology was not such a strong science as it is now. And then very few reports during the last 3 to 4 decades from both India and Ethiopia, because even though the cultivation continues, it is taken in smaller amounts, the diet is more diversified, there are more public health programs in terms of PDS distribution of grains and other things, so the disease to a great extent has disappeared. Now, despite animal models, as I said, the disease is still an enigma. Large doses are required for oral feeding even in animals, and symptoms vary widely. It is a glutamate receptor agonist, an excitatory amino acid, but does not significantly bind to glutamate receptor. That's something which is again a gap in our knowledge. But other mechanisms of damage have been worked out. It produces oxidant damage, inhibits glutathione synthesis, which is important to overcome the oxidant damage. VEGFs are involved in neuroprotection and survival of motor neurons and are reduced in lumbosacral region in Lathyrism. This is an important thing which has been done in the experimental systems. It contains homoarginine and it can, if high homoarginine is present, it leads to excess nitric oxide production and damages the mitochondrial DNA. But if it is much lower, as it is in the 50 grams of pulse, that it is 2 grams per 100 grams, so it will be less than about 0.5 grams, that would be a useful thing for a human being. It inhibits tyrosinamide transferase, enhances DOPA and 6-hydroxy-DOPA, which also cause neurodegeneration. So some of these things are the new mechanisms of toxicities which have emerged from the recent studies. And in sharp contrast to animals, humans detoxify ODAP. So those who develop the disease may not have this capacity. This is something which has not been worked out. Deficiency, I told, of sulfur amino acids exaggerate the disease. Estrogens protect against mitochondrial respiration. Therefore, laterism is not common in women. If at all, if it occurs during the pre-pubertal stage or after menopause. There is a hypoxic inducible factor. This has been worked out only in neuroblastoma cells. It's a primary adaptive response to lowered oxygen concentrations. And ODAP increases this, and therefore it stabilizes the response. This is an exciting application of ODAP to overcome hypoxic events. So the beneficial effects, can it be a functional food, is the next question which needs to be answered. Though the research is not being continued in India, in other countries, particularly in Europe, it's being done. A symposium was held sometime, I think in 2003 or 4 in NIN, where several talked about it being a PKC, protein kinase C activator, which is neuroprotective and neurotropic. Contains homoarginine. I talked about 2 grams per 100 grams of seeds. It is a substrate for nitric oxide synthesis and benefits vasculature, that is, dilatation, vasodilatation. It is also a signaling molecule in cardiac and cerebral metabolism, and lower circulating homoarginine is a risk factor for sudden cardiac deaths. This has been done in human subjects. Commercial performance boosting products contain arginine. That's because it supplies more oxygen. Therefore, can lathyrus be considered as a functional food for hypertension is important, and it is also associated with increased bone turnover. The deficiency is associated with increased bone turnover. So can it be used to alter the bone mineral density? Can it improve the bone health? Are questions which have to be answered. Arginine is also a secretagogue of insulin, and pancreatic arginase destroys arginine very fast. But homoarginine is not a substrate for arginase, and hence it could be considered for several health benefits as well. So more work is required for it to be considered a functional food in terms of the amount of pulse which has to be taken, the amount of homoarginine which is required, and how long should be the exposure. So these are the questions which I think perhaps will be answered maybe in the next few years. Now let me go to the next subject, fluorosis. Fluoride halogen is an electronegative element and highly reactive. It's a cumulative poison resulting in teeth, bone and joint affliction. I don't think I need to go into the details. It occurs in sedimentary limestone or sandstone and igneous granite rocks, and therefore the weathering of these minerals along with volcanic and fumaroloric process, that is the fumes, will lead to higher fluoride levels in the groundwater. It is essential, it is important for mineralization, but has a very narrow margin of safety. It occupies the 17th position in order of abundance. I'm sorry. 17th position in order of abundance in nature, and it finds its way into seawater, underground water and vegetation. Several factors, of course, determine the solubility, and that's an important thing, and that's why there is a belt which extends from Iraq to Iran to China to India. In addition, environmentally, fertilizers, pesticides, particulate industrial emissions, etc., add to the fluoride in nature. Construction of reservoirs, dams, alter the subsoil and mineral composition. Actually, the subsoil pH is altered, the uptake of several minerals are different, and that results in excess bone abnormalities as the accretion and resorption of bone, altering the homeostasis of mineral metabolism. So this geo-environmental health issue exists in India. It has been reported from almost 24 countries. From 1931, the reports exist. China, Japan, Sri Lanka, US, Germany, Southern Arabia have reported the occurrence. In India, 62 million suffer from fluorosis, including 6 million children in 18 different constituent states and union territories. The principal source of fluorine is, of course, drinking water. But food also cannot be eliminated, such as sea fish, cheese, tea. They also contain high amounts of fluoride. I will show you another slide which gives all the fluoride levels in different foods. Cereals and pulses grown in fluorotic area are also high in fluoride. Of the 85 million tons of fluoride deposits on earth, almost 12 million are found in India. So India is really endemic for fluorosis. And the recommended level of fluoride is in India is 1 milligram per liter, or 1 part per million, whereas WHO recommends 1.5 milligrams. India being a tropical country, we drink more water, and therefore we have recommended much less. Now that's I don't think this slide is very clear. I'll skip it because I have imported it from a website. Dental fluorosis is the commonest thing which is encountered in all the areas where fluoride levels are higher. I'll show you a typical picture of that. These are the various grades, and now there is a community index of dental fluorosis available according to WHO, which is used by field workers to measure dental fluorosis. Then skeletal fluorosis, there are a spectrum of manifestations. It's a very slow and progressive but dynamic disease. Characterized by the musculoskeletal dysfunction, arthralgia, arthritis, stiffness of spine, narrowing of intervertebral foramen, compression of spinal cord, osteosclerosis, interosseous membrane calcification, typical histological findings of fluoroapatite formation, and is toxic to the osteoblasts and repair mechanisms in the bone, which leads to abnormal bone formation, and fracture rates are very high. So a combination of osteosclerosis, porosis, malacia, and exostosis are very common. Of course, type of bone abnormalities will depend upon calcium intake and other trace elements. I'll come to that point later. The prevalence in Indian states vary from 14 to 40%. Rajasthan, Gujarat, and Andhra Pradesh are the affected affected states, whereas Punjab, Haryana, Madhya Pradesh, Maharashtra are moderately affected. Tamil Nadu and Karnataka, West Bengal, Uttar Pradesh, Bihar, Assam are mildly affected. It depends upon how much of fluoride is there in the water. That's a typical picture of a victim, extremely stiff. There are several other risk factors as well. In certain countries, for example in Vietnam and also Thailand, water fluoride is low, but still they get fluorosis because the food fluoride is very high. That must be from the soil, and we really do not know what exactly is the source. Maybe there are some granite stones and things, and there is volcanic eruptions which produces the fluoride. Sorghum, that is jawar, most likely to, those on sorghum are most likely to develop the severe manifestation. Silicate is also responsible for fluoride retention. The millets contain high silicate consumed in endemic areas. And dietary deficiency of protein, calcium, vitamin C exaggerate the fluoride toxicity. Parboiled rice intake increases fluoride burden because the water used is high in fluoride. And construction of water reservoirs, as I have said, alters the pH of subsoil water, promoting the uptake of trace minerals. And several levels of fluoride are high, serum levels of fluoride are very high in these areas. And there is a very typical manifestation of fluoride toxicity called the genu valgum, which occurs in adolescents between the ages of 10 to 15. And endemic zones are China, South Africa, India, Tanzania, etc. And it is again common in jawar eaters. Dental and skeletal fluorosis are common. Osteoporosis of limb bones is coupled with hyperparathyroidism, and serum fluoride is very high, while calcium, phosphorus are normal, they are not reduced, and alkaline phosphatase is higher. And some of the fluoride deposits in this bone have been looked into who have undergone reconstruction surgery in our own nutrition unit hospital in Usmania Hospital, where we were working on this, and the fluoride levels are very high. What is interesting is to observe that copper content in this bone is only 35% of the normal values, and therefore there is a poor collagen in the bone, which increases the hydroxyproline excretion in the urine. Serum levels of growth hormone are high, PTH is high, and in the genu-valgum cases, the 25-hydroxy vitamin D is high, while thyrocalcitonin is low. This particular aspect has never been explained because no one measured 125-dihydroxy vitamin D in those days. And as I said, on sorghum intake, fluoride retained is much higher. There is a high molybdenum intake coupled with low copper, which precipitates the genu-valgum, and breast milk content of copper was found to be very low in these areas. A negative correlation exists between copper in drinking water and prevalence of genu-valgum. These are some of the new aspects of the fluorotic disease. This, of course, is also now almost 20-30 years old. This is the typical picture of all adolescent people are being affected. Now, more recently in Bihar, very young children were found to have genu-valgum, but this is not associated with osteoporosis, but more of osteomalacia with lower vitamin D. This is very characteristic in young children whose calcium intake is extremely low, and the phosphate intakes, particularly from potatoes, have been found to be very high. So there are different manifestations depending upon the various situations in relation to these levels. Now here is the 25-hydroxy D3 levels. This is in the endemic area, and age-matched endemic area who are not having genu-valgum, it is low, and from the non-endemic areas is much lower. But why is it high? Why, even though the parathyroid is also high, it should actually, vitamin D should be lower, but it does not occur in genu-valgum. Probably there is a block in the conversion of 25-hydroxy to 125-dihydroxy vitamin D. Now, trace metals, I have said silicon, molybdenum and copper. I have already talked about it. I don't want to go into the detail. They are all essential for normal metabolism of bone and connective tissue, but excess with the synergistic activity of fluoride, there is on the mineralization, enhances the toxicity. Similarly, molybdenum in the fluorotic areas is very high and promotes retention of fluoride in the bones. This occurs only very near the dams or water reservoirs constructed, because the uptake by the plants is totally altered. And when fluoride and molybdenum higher, it promotes copper excretion, and you get a copper deficiency. So you get a picture, a complicated picture, and these are some of the values. I don't think I will go into the details. I have already summarized. Okay, let me look at the total dietary intake of fluoride and silicon in the fluorotic area as compared to the normal areas. Urban and rural itself, there are some differences because of the water resources used are different, but the diet does not differ. In urban area, it's silicon only 7.5, but the rural it's higher, but it's much higher in case of a fluorotic area. That's because of the subsoil water alteration. Effect of parboiling is very clear whether you use the different varieties of rice. The Hansa and Sona have been used, and they are found to be very high. So bone mineral density is higher, but the bone quality is poor, and therefore particularly in the forearm you can see the density is reduced, and that's where the fractures are extremely common in the fluorotic areas in almost all the age groups, particularly the long bones. Lower end of the forearm and lower end of the tibia are the ones which are affected. There are other changes in the other organs and tissues, soft tissues. I don't want to go into the details, but muscle and nerve pathology are important. The muscle pathology is always secondary to the nerve pathology because it compresses the spinal cord. Tamarind, actually this originated from Dr. Sri Ramachari in All India Institute of Medical Sciences, Delhi. Tamarind binds fluoride, and so several experiments were carried out, including human and animal experiments. And healthy young boys were put on 10 grams of tamarind per day, and they found that there is an increased urinary fluoride. It's significantly different. I have not put the SDs and HCs. There is also excretion of zinc and magnesium have been reduced in such a situation, which are beneficial for the bone. And therefore, the urine actually becomes alkaline and reduces excretion of zinc and magnesium. So, alkalinity is an important factor. So, this is which I have already spoken. I don't want to go into the details of it. There are several defluoridation techniques, Langonda, Prashanti technique and ICOH technique. This is the International Community for Oral Health ICOH technique. All of them have been practiced. All of them have some defects or the other. It's not that easy to keep them going. The domestic and community filters are available. This worked very well in the Nalgonda district of Andhra Pradesh. But even the domestic and community level Nalgonda district NEERI technique, that is National Environmental Engineering Institute, has also come out with that. It did work, but the domestic one worked much better than the community levels. The domestic is very simple. Take 40 liters of water, add alum and bleaching powder and lime, allow it to settle for about 2 hours, and then you stir it 10 minutes. It's a flocculation precipitation method, and then the water, supernatant water, is drained for use. This was found to be effective, but ultimately for a prevention of fluorosis, you need to supply them, the population at risk needs to be provided water which is about 0.1 to 1 ppm fluoride safe level. And how can you do that? You can do from supply of surface water from rivers, canals and dams. And that's the best way by which you can prevent fluorosis, because both these diseases, whether it's laterism or fluorosis, you do not have any treatment for it. And defluoridation of local water by appropriate method can be practiced. There can be improve the nutrition situation and avoids food high in fluoride content by the population at risk. Now, avoiding indiscriminate use of fluoride-rich pesticides and fertilizers are as important because they contaminate the environment. Parboiling paddy in safe water supply is an important thing. And more recently, rainwater harvesting, which is anyway important for us to do because of water conservation, is extremely useful because this can be used for drinking and cooking purposes. So that's exactly what perhaps I wanted to say, not more, not less, because I'm the last speaker. If there are any questions, I'll answer them. Thank you all for your patient listening.
Thank you, ma'am. Just one question I would like to ask. Does this home water treatment like reverse osmosis and filtration, are these in some way effective in removing?
Reverse osmosis can take out the fluoride, but it's very expensive. It's not an easy one. At a community level in a village.
No, but at this home appliances, they are available, RO
home
system.
appliances now are available, which the reverse osmosis are supposed to take out all heavy metals. Fluoride is also one of them. But that's all expensive procedures. No, it cannot be practiced for community practice. It's not possible. Yes, please. Yeah, tamarind actually, it mops up the fluoride. And whatever tamarind is, whatever the tannic acid and other constituents are absorbed, it also leaches out the fluoride in the urine. That's how in children who are being given 10 grams of fluoride, 10 grams of tamarind per day, urinary fluoride excretion is higher. But it is not a practical, you have to give for an established disease, you have to give it for years. You can only prevent for children, but how much of tamarind you can take is not very high. It has been found that it is one of the useful things, but they are not practical measures. I won't say, the only practical measure is supply of water from surface waters and not the underground water, because the underground water is always higher in fluoride. And earlier some people came out with the view that boring deep wells, you will not find fluoride, but even there is fluoride. In the fluoride belt, fluoride is there everywhere in the underground water. No, you see, why I called, I'm sorry I did not perhaps miss that slide. Fluoride is a friend because in US, wherever, when it is less than 1 part per billion or 1 part per million, that 1 milligram per liter, it is, caries are very common. So US and in some countries in Europe have used fluoridation as a public health measure to prevent dental caries. It's also given for osteoporosis because it produces sclerosis. It's given for autosclerosis in ENT practice. So it is a friend, but it's the dose which makes the big difference.
Thank you, madam, for your excellent presentation.
Thank you, ma'am. Now may I request Dr. Pradeep Bhatia sir to facilitate Dr. Kuldeep Singh sir. Kuldeep Singh. Thank you, sir. Now may I request Dr. Pankaja Raghav ma'am to felicitate Dr. Praveen Sharma sir. Thank you, ma'am. Now may I request Dr. Pradeep Bhatia sir to felicitate Dr. Kamala Krishnamurthy ma'am. Thank you, sir. Thank you, ma'am. Now may I request Dr. Sneha Ambani ma'am to felicitate Dr. Pradeep Bhatia sir. Once again, I request Madam to felicitate Dr. Pankaja Raghav, ma'am. Thank you, ma'am. All the delegates are requested to fill the post-test assessment and symposium evaluation forms. After a break of 15 minutes, all of you are requested to rejoin us for the valedictory function.